Here is the Chapter 22 lab where you're going to diagnose patients using their white blood cell counts. These are the papers that are in your binder. So it begins with some background information. Make sure you read this. It explains about the white blood cells and it helps you to understand what's going on in this lab. Then here are a list of diseases, and the ones you're going to diagnose are from this list. So in this lab, we have 5 patients. We have 5 bottles of blood samples here. And notice each one is clearly labeled. This is patient 1, this is patient 2, and so on. We have patients 3, 4, and 5. And we're going to count their white blood cells, and then we're going to figure out which condition each patient has from this list. So notice how the list, like for example, autoimmune disease, neutrophils and lymphocytes are decreased. So when you do the white blood cell count, if you see someone whose lymphocyte or neutrophils and lymphocytes are low, then that might indicate an autoimmune disease. Now you can't diagnose them just from the white blood cell counts. You also have to look at the patient information. So the white blood cell counts are one clue. When it comes time to diagnose your patient, you also have to look at this information, right? Like whether or not they have a fever, if they have nausea, what the other signs and symptoms are. And you put all of that information together to figure out which condition your patient has. Now, in order to know what's abnormal, you have to know what's normal. So this chart shows you the normal ranges. So you're going to figure out your patient's white blood cell counts and you're going to compare that to what's normal. So that way you know if their neutrophils and lymphocytes are low and so on, the other clues here. Okay, so this goes through and it explains how to do the white blood cell count. In these bottles of blood, and remember, this is not real blood, this is simulated blood, but in these bottles of blood, when you take blood from a patient, it's too concentrated. Like if you tried to count the blood cells, there's just so many there, it would be an overwhelming task. So we dilute it. Diluting it makes it easier to count. So this is what we use to dilute. So I'm showing you this picture and your instructions. This is the actual device. So what we do is we put some blood in here and we put some water in here. So I have a beaker of water. And you get that blood and water into this little chamber here. You can kind of see this expanded part. Then once you have them in there, you cover the ends of this and you shake it up to mix them, so that then you have a diluted sample of blood in that chamber. And the way you do this, you have at the other end of the hose here, you have a syringe. So you just pull the top up to bring it in and you push it down to squirt it out. So we're going to pull some blood into here, then we're going to pull some water into here, then we're going to shake it to mix it up. Then, because water was the last thing you pulled in, the whole tip here is going to be full with water. So first we pull the blood in, and you have blood in the tip here. Then you pull the water in, then that water coming in pushes the blood up into the chamber, some of the water goes into the chamber. You mix that up, you have your diluted blood in the chamber. Then we have to clear just the plain water that's here. So then you squirt about 5 drops out onto a paper towel. That way, the diluted blood mixture from the chamber is now down at the tip. Once you do that, you're ready to load it into the slide. So this is a special type of slide called a hemocytometer. And you can't see it when you just look at it regular. But on those metal parts in the middle, there is a grid etched in there. And that grid helps us count. So then what we would do next is we put our cover slip on here, and you make a drop of the diluted blood form at the tip, and you touch it right there to that base of that triangle. And these slides use capillary action. So when you touch the drop there, capillary action will suck that drop of blood in, and you will then have your drop of blood on the grid. So they have a picture in the instructions here. When you put that under the microscope, this is what the grid looks like. Now remember, this is what it looks like under the microscope. So when we look at it sitting here, you can't see the grid. You have to put it under the microscope because the grid is very tiny, but it's etched in there. Now the letters A, B, C, and D, those are not on the actual slide. They just put those on here to make it easier for you to understand the instructions. But on the microscope, it will look like this. And I have included in your soft chalk a photograph of what it actually looks like under the microscope. Then you count your white blood cells. So the white blood cells are just going to look like little circles. They're not going to look like the cells in the photographs in the picture, because in order for them to look like, or the photographs in your book, in order for them to look like that, you have to stain them, and you have to go through a different process of putting them on the slide. And it's the process that was covered in chapter 18, how you make a smear and you stain it and you dry it and you look at it under the microscope with a different procedure. So with this procedure, all they look like is circles. And sometimes on your slide there might be dust or debris, and the way you can distinguish is the blood cells are round. If you have a little piece of dust or something on there, it's going to be kind of jagged and irregular shaped. So when you see these perfect circles, those are your blood cells. And you go through and you count them. And the purpose of the grid is it just makes it easier to keep track of where you're counting. So you start with this square, and you would count how many are in this square, then how many are in this square, and so on. And that way you keep track of which ones you've counted and which ones you haven't if you follow along through the squares. Then in this chart is where you fill out your counts. So remember here, you have 4 different counting areas. So you're going to take your microscope and you're going to zoom in on this one. And you're going to count how many white blood cells are in that area of boxes. Then that number gets written here, counting area 1A. Then you move your slide so that now you're zoomed in here on counting area B. You count those white blood cells. You fill that in here, counting area 1B. Then you repeat that process with C and D. Over here, notice it then has a column for counting area 2. Notice there's 2 sets here. You have this side is counting area 1. Then you have over here is counting area 2. So then you would move the slide on the microscope over to counting area 2, and you would repeat that whole process. You would count the white blood cells in A, B, C, and D, and you fill that in counting area 2. Then you get your average total. So in science, you always want to do replicates. You don't want to just rely on one count. You want to do multiple counts and then average them together. That gives you a better picture of the data. So then you take your counting area 1, your counting area 2, and you average them. Then we have to take into account that we diluted this. So we just counted diluted blood. We want to know how many white blood cells are in the actual patient. So we have a math formula to help us figure that out. So you take your average count, you multiply it by 20, and you divide that by 0.4. So that's how we take into account the dilution that we did when we were mixing blood and water here. The count you just did are a count of diluted blood. You do this math formula to tell you what the actual count in the patient's blood is by using your diluted count and doing the math to work backwards to the patient's actual count. Once you have all of those counts done and you have the patient's actual blood cell counts, then we need to start looking at the differential. So you need to know how many of those white blood cells are eosinophils, how many are neutrophils, how many are basophils, and so on. Because remember, back here, where you're diagnosing your patient, you have to know the individual types of blood cells and which ones are high and which ones are low. So in a lab, you would do another count of the patient's blood where you would go through and you would actually distinguish the cells. They wouldn't all just look like individual circles. They would look different from each other, and you could say, like, oh, 15.5% of this patient's cells are eosinophils, and 34% of them are neutrophils, and so on. Well, in this lab, they did that for us. So the makers of the lab kit already are telling us that patient 1 has 15.5% eosinophils, 34% neutrophils, 4% basophils, and so on. You have to now figure out how many eosinophils patient 1 has. Well, let's suppose patient 1 has 2,000 white blood cells altogether. If 15.5% of those are eosinophils, you just take 15.5% of 2,000. So depending on how your calculator works, if you have a percent button, you just push 15.5% button times 2,000. If you don't have a percent button, you just take 0.155, because to transfer from percent to decimal, you just move the decimal 2 places, times 2,000. So we'll go through that together. We're going to say that patient 1 has 2,000 white blood cells total. That's the number we got from our counts, and then doing the math to take into account the dilution. We know that 15.5% of those are eosinophils. So you can take 15.5, then notice I have a percent button. I push that. Now I have 15.5% times 2,000. That gives me 310. So I would write 310 in that box right there. So I know patient 1 has 310 eosinophils. If your calculator does not have that percent button, all you do is move the decimal place 2 points. So we have 15.5%. Well, if you move the decimal to the left 2 places, we would put 0.155. Then take that times 2,000. And again, we get 310 for the answer. So you go through and you fill in this whole chart doing that. Then once you have that chart filled in, you're ready to diagnose your patients. And this is where you go through for each patient. We go back to that chart we had of the normal counts. So right here is your chart of normal counts. And you compare. So patient 1 has 310 eosinophils. So eosinophils should be 50 to 400. So that is within the normal range. So then we would look at their neutrophils and we would compare that and see if it's within the normal range. And an easy way to do this is to get a highlighter and go through, compare each patient's cells. If it's abnormal, highlight it. If it's normal, just leave it alone. Then you can see which counts are abnormal. Then you come up here and you compare what your patient had as abnormal to what's listed for each disease as abnormal. And remember to also take into account the patient's other signs and symptoms, and you diagnose your 5 patients. You come up with which condition you think they have. And I'll give you a hint, you have 5 patients, 4 of them have a medical problem, 1 person comes back normal. And that happens. Just because you test somebody for a disease doesn't mean they're going to have it. It happens all the time that you test people and then they come back that they're actually fine. So 1 person's going to be fine, 4 people are going to be sick. After you've done that, then there's just some other general questions about the immune system and different types of blood cells. When you get to this one, the matching, Each cell will have either 2 or 3 letters that go next to it. Okay, then you just answer the other questions, and then you're done with this lab.